give a short introduction about Purolite LifeTech, the new platform of enzyme carriers and media for upstream and downstream processing in bio-applications. Just an overview about Purolite. So Purolite is not new in the market. It's a company founded in almost 30 years ago by two brothers, Steve and Don Brody. Purolite at the moment is the second largest supplier of ion exchange resins in the market, and they are leader in this industry. Purolite has a great level of technical excellence and dedication in the production of ion exchange resins that no other companies provide. So innovation is the key of success of Purolite, and we have 5 research and development centers that are coordinated from our R&D Center of Excellence located in UK. And here is also present the R&D manager of Purolite if you have any question. So the key of excellence of Purolite is our support to customers. So we have more than 40 sales offices in different countries. We have a large supply chain network designed to support customers really close to their needs. So we have 3 research and development centers in Europe. Then we have a research and development center in US and 1 in China, where we have 3 manufacturing sites, 1 in US, 1 in Romania, where we manufacture GMP products and products for pharma, and in China. So our production capabilities are, of course, on multi-ton scale. Just to give you examples of the variety of products that we provide in different industries. So for food, we have nice examples of resins for color removal from the refined sugar, so in the production of sugars. In the groundwater remediation, we have nice products for removal of contaminants as chromium, uranium, or perchlorate. We are very strong in the hydrometallurgy, so we have resins specific for the extraction of gold, silver, or other precious metals and rare earths. And in pharmaceuticals, so we have some APIs that we produce as products for decreasing the cholesterol level, for the potassium level regulation in the blood, and tablet disgregants. So starting from this excellence and experience in resin manufacturers, we developed the new business unit which is called Purolite LifeTech, so focusing on all the bio applications. So moving to the biocatalyst, solid phase synthesis, and purification, and all the platform for chromatography in the upstream and downstream processing. The main area where we focus with Purolite LifeTech is affinity chromatography, a wide platform of resins for peptides and oligonucleotide production and purification, and of course biocatalysis. So we are the company that offers the widest and portfolio of resins for all the biotech applications, and we provide standard and also customized solutions for all our customer needs. For biocatalysis, of course, we understand that making a good biocatalyst, so an enzymes that works in industry, is not a matter only of having a good enzyme with the right selectivity and stability, but the carrier plays a key role in obtaining a very strong and robust biocatalyst. So we can play with the chemical properties. We have different kind of matrix, acrylic, styrene. We can play with the physical properties, so surface area, porosity, kind of ligands, and the mechanical stability. So industry is moving to large and large scale applications, and we need to provide resins that are robust, that can be used many, many cycles. So having an efficient immobilization method will give a high productivity, so a lot of products per unit of enzyme consumed. Of course, there are some advantages and disadvantages, but Here I'm not, I don't want to convince you about the benefits of enzyme immobilization. Of course, there is an easy separation. The enzyme is immobilized on a carrier in a physical bit, so it can be easily separated by separation, and you reduce the cost of downstream processing. The platform that we have for enzyme immobilization exploit the chemistry that we have with acrylic, with styrene, with the porous and macroporous resin. So we have some resins as the macroporous for absorption of enzyme. This is a technology that is unique in Purolite. So you have resins with a very high surface area and a very high porosity. So very good for large enzymes, but also for all the process where the kinetics are a key factor limiting the reactions. We have also different kind of resins functionalized with octadecyl. So here you exploit the matrix, which can be styrene or acrylic, and then you have different pendants, different loadings on pendants, so C18 on the resin, that favor the absorption of the enzyme. And then we have a panel of resins for covalent immobilization, so the amino resins, where you need to pre-activate with glutaraldehyde and the epoxy. These are the standard products, but we can customize functionality, so we can design the functional group that you need for your process. Porosity is something that we can tailor, so we can go from less than 50 Armstrong going up to 2,000 Armstrong. Of course, we take care about creating a polymer that has enough mechanical stability to support a strong industrial process. And of course, the particle size. So our standard particle size are 150, 300. I will call the F grade, typically for pharma, and 300, 700 micron for the M grade. But we are also developing these carriers on a monodisperse range, so you will have only one size designed specifically for your application. Just to give you some examples of advantages of ECR resins compared to other resins. So this is a classical Novozyme 435. It's a Absorb-Cal-B on a levated product. Here the immobilization is by adsorption, and we compare different results on acrylic and styrene resins using the same protein loading. Sorry. So you can see that compared to the levated VP, compared to the levated VPOC 1600, we have some carriers as the 1090, 1091, which are macroporous resins, where you get more activity than on the levated resins. And we have the new developed ECR 1030, which is a microporous resin, styrene absorbent, that can be designed in different particle size and shows very strong mechanical stability. And then we have the ECR 108806, which is an octadecyl resin. So here you exploit the absorption, but the interaction with the octadecyl groups, and the activity that we can achieve is almost 20,000 units per gram. Another advantage of the ECR resins, apart from activity, is the mechanical stability. Here we compare our resin in blue and the levated resin in red. So this is a test that measures the formation of fines during magnetic stirring. So you can see that the blue doesn't form too many fines, so it's very, very stable mechanically. And after 3 hours mixing with the magnetic stirrer, you can see that our resin doesn't form any fines compared to the levated. So this is very important when you run batch reactions. Another test that we did for checking the mechanical stability of our resin is the Russian ball mill test. It's a special test that we run for mining. So you mix the resins with some sand, and you mix and you verify the formation of fines. It's a very strong method, but the comparison, you can see that after 6-hour test, you have almost complete recovery of the ECR 1030, whereas for the lipozyme, you have 70% of remaining beads and 30% of formation of fines. There are also some resin designed for covalent immobilization, so you can covalent immobilize Cal-B, and you can work in biphasic system. So the comparison with an epoxy and hydrophobic resin is clear. So if you have an hydrophobic resin, so with some styrene in the matrix, you have better interaction and better activity. For chromatography, we have a wide panel of resins. For preparative and analytical chromatography, we go from 3 micron, so the typical HPLC grade, to 10, 15 micron for the process and polishing steps of small molecules and synthetic biomolecules, up to more than 75 microns. So here we have all the range of synthetic. This is not silica. The advantages of synthetic resins are the high pH stability. So you can run chromatographic separation also and sanitization steps without any disruption of the silica. This is just a panel of showing the panel of the resins that we have available. So we have a panel of resins with ionic exchange functionality, so functionalized with quaternary amine or sulfonic acid. So here you have the anion and the cation with different particle size. So we have the functionalized resins from 15 to 50 and more than 200 microns. And for hydrophobic interactions, so the hydrophobic interaction and the reverse phase, we have a huge panel of resins, the 3, 4, and 5, 81, which are typical HPLC. Then we have the 82, which is more porous, where we can have the scale up because we can go from 5 to 10 to the 15 micron. And we have the PCG line, which is offering different particle different porosities. So 1200 means 1200 Armstrong, so it's a very porous matrix for large biomolecule, 900 and 600. So we offer all the panel of resins for all the purification of biomolecules. Just to show you here the technology that we have for producing this kind of resins, this is polystyrene in 15 microns. This is the PCG 1200, so you can see the high porosity. And this is the polymers as they appear after polymerization without any sieving. So we have a technology that allows us to produce really monodispersed bits. And the mechanical stability of these resins against back pressures is very high compared to other materials. Yeah, you can see here the back pressure goes up to 250 bars, so, which is very good for the prep HPLC. Here are some examples of purification of insulin using different kind of adsorbents. So here is the PCG 1200, and you see here the scale up is very good from 15 micron to 35 micron. And here you see the comparison with some existing resin. This is a silica, and these are synthetic resins. Yeah, thank you.